Hi, all. This is Catherine Cummings, and I will be presenting on the disease which is known as chronic wasting disease for the FOWS 7200 disease ecology. I'm a current PhD student who is looking at the human dimensions of chronic wasting disease. For the rest of my presentation, I'm going to refer to chronic wasting disease as CWD just for simplicity. And I'm going to begin this presentation with an overview of CWD and its characteristics before discussing my final paper topic for the course. I'm also affiliated with the Conservation Governance Lab if anyone's interested in looking that up. As for a bit of background, I wanted to discuss the pathogen or disease-causing agent of this disease. The pathogen of CWD is an infectious prion, which is a protein. It's caused by an infectious misfolded protein isoform of the host protein, which if you see this figure right here, we see the normal protein being transformed into an infectious protein or prion. The disease, which is caused by this infectious prion, is known as chronic wasting disease and typically affects cervids. So CWD is a 100% fatal neurodegenerative disease that affects members of the Cervidae family, including deer, elk, moose, and caribou. The disease is naturally occurring and is caused by a misfolded prion, as I stated before. Due to the fact that CWD is caused by this infectious protein, there are no preventions, treatments, or cures for the disease. CWD is characterized as having a long incubation period, which ranges anywhere between 18 to 24 months, where an infected animal appears healthy, but is carrying and spreading the disease to other healthy individuals, as well as throughout the environment. The infectious prions are able to replicate inside of this infected animal and, like I said before, spread it to other cervids in the area. As for a bit of history and the distribution of CWD, CWD is part of a family of diseases called transmissible spongiform encephalopathies. So these transmissible spongiform encephalopathies have similarities, and we see them in scrapie in sheep and goats, bovine spongiform encephalopathy, otherwise known as mad cow disease, and Creutzfeldt-Jakob disease, which affects humans. As for some history, CWD was first discovered in a captive research facility in Colorado in a mule deer herd in 1967. Elizabeth Williams, who you can see up above here, was the first person to identify the disease, and she was a graduate student at the time. She noticed clinical signs that developed that were similar to scrapian sheep, but were a little bit unique for the deer. As for the distribution of CWD, CWD has been identified in the United States, Canada, Norway, Finland, Sweden, and South Korea. But here in the US, CWD has been positively identified in 30 herds in either captive and or free-ranging herds. And you can see that on the map down below. As for symptoms, CWD is typically asymptomatic in the cervid. I might refer to it as deer. So if I do, just know I mean cervids in general, for approximately 18 to 24 months. So that long incubation period, which I talked about before. When symptoms do arise, they can include, but are not limited to, extreme weight loss or emaciation, stumbling, excessive drooling, excessive urination, drooping head or stature, and other neurological symptoms. I just want to state that if a cervid is displaying these symptoms, it does not mean that the animal is CWD positive, since these symptoms are pretty similar to a lot of other diseases that deer can get. So a CWD diagnosis should be determined by a diagnostic testing, which I'm going to talk about a little bit further on. As for transmission of CWD, infectious prions can be transmitted both vertically and horizontally, which is the same for all transmissible spongiform encephalopathies. Vertical transmission, or the passing of infected prions from mother to fetus while in utero, has been documented in both muntjac deer and elk populations, but it's not typically seen in the wild. horizontal transmission occurs from either indirect or direct contact with infectious materials. So indirect contact with infectious materials can occur through the ingestion of plants that have been contaminated with these infectious prions, or through direct contact, which can include coming into contact with bodily fluids such as feces, urine, saliva, and blood that can be shed by an infected deer through the duration of their incubation period. These infectious prions can also live indefinitely in an environment, so it is pretty concerning that they are able to be in this environment and spread to other healthy cervids. As for spillover, I just wanted to touch on this a bit as it relates a little bit to my topic. So currently, susceptible species of CWD only include cervids. There's been multiple scientific studies that have tried to identify if CWD can cross species barriers, but to date, no studies have identified the major mode of transmission for CWD or the associated factors that might have influenced the pathogen's preferred mode of transmission. So I have a couple other species up here, which have all been tested to see if they're susceptible to CWD, but typically these scientific studies occur either in vitro, in a lab, and most of the time, there's a lot of conflicting results. So one study will say that spider monkeys are susceptible, while the other study says that they are not. So this is just something concerning that we need to keep in mind, especially when we're trying to convey this information to the public. As for testing, there is only pretty much post-mortem testing, which is approved, and this happens after either an animal is harvested by a hunter or maybe hit on a road and is roadkill. But typically, we can only see post-mortem testing for CWD. There's currently no live diagnostic testing that is approved that hunters or state agencies can use with clarity and approved by the government. So for this post-mortem testing, lymph nodes or brainstem must be collected in order to be tested. So state wildlife agencies really rely on hunter-harvested samples to be able to track the prevalence rate of CWD throughout their state, and especially in localized areas where CWD is known. Additionally, state agencies also collect roadkill and high-value samples, so deer that appear to be sick or have symptoms similar to CWD throughout the year. For diagnostic testing, like I said before, the only approved methods right now are postmortem, and there's only 2 approved methods which a lab can run to see if an animal does have CWD. So we have immunohistochemistry and IHC, or enzyme link immunosorbent assay and ELISA. CWD can also have a definitive diagnosis based on Western blotting, prion misfolding, cyclic amplification, and real-time quaking-induced conversion, but these methods have not been approved yet, but they could be in the near future. So you might be asking yourself now, why should we even care about CWD? What is the major implications of this? And why are we putting so much time and effort into research about CWD? For starters, studies have suggested that once prevalence rates reach 28% or higher, we can typically see population declines in both population size and herd health of cervid populations. Additionally, in areas where CWD has been present for a long period of time, prevalence rates can range anywhere from 10 to 25% in these localized areas. These high prevalence rates can influence hunters to not purchase a hunting license. Thus, this really hurts state wildlife agencies' funding mechanisms. I'm not going to get into the nitty-gritty of funding mechanisms for state wildlife agencies, but if you're interested, just feel free to reach out to me, and I'm happy to talk about it. It's a topic I really love. But for instance, we saw that in Wisconsin, after CWD was discovered, there was an 11% reduction in hunting license sales after CWD was discovered. This meant that the state faced an 11% reduction in the amount of funding which they had to mitigate the disease, and they also had to allocate funding from other conservation efforts to disease mitigation, which included testing, as well as maybe some mitigation methods like sharpshooting or reducing herd populations. So with CWD spreading throughout the United States, we should be concerned that CWD could have negative health implications on cervid populations and possibly down the line other species if the disease is able to cross species barriers. So for my project for this course, I know this wasn't really required to be added on, but I just thought it would be something interesting to add on in case anyone's interested in talking about it with me further down the line. Just feel free to reach out. But to date, and to my knowledge, there's no scientific studies which are a review of all of the existing literature of the current state of zoonosis of CWD. There's been extensive studies that have tried to determine the susceptibility of other wildlife species in regard to CWD. But like I said earlier, there's a lot of contradictory results that have been published. So there's some studies saying that certain wildlife species are able to be susceptible, while others completely say that they're not able to be susceptible. I intend to gather all of these studies that have been published and include them in one document so current and future wildlife agency employees and wildlife health specialists can determine if CWD actually poses any risk to their surrounding wildlife or humans in the area. While CWD has not crossed species barriers in the wild that we know of, other prion diseases such as bovine spongiform encephalopathy have been able to become zoonotic. So I might also include the similarities of bovine spongiform encephalopathy and CWD in this paper I write to show that there is a zoonotic potential of CWD. So as you can see above, I'm just showing you an example of how I'm going to write up my results. So I have the article name, the species that was used, the type of CWD material that was used. This also ranges from study to study. The route, so some studies just have an animal come into contact with it itself, or they have some type of inoculation either orally or through the live species brain. I want to see if CWD transmission occurred, the length of time until symptoms, and any interesting notes that might come up. This table might change, but it's just something that I think is really interesting and important to look at. So with that, if anyone's interested, my contact information is here. It's really been a pleasure taking this course and reading everyone's blogs. And like I said before, if anyone has any questions or wants to discuss this any further, please feel free to reach out to me. And thank you for listening. And here's just a short list of some references I used. So thank you.